So in this next lecture segment, we're going to consider the rules that govern how molecules will or will not be able to cross over a cell membrane. So we're going to talk about some basic rules of chemistry and physics, and then how it applies to the movement of molecules across a membrane. So a few definitions. Passive transport, when you see the word transport, that means something's being moved across, and in our case in biology, moved across a membrane. So passive transport is defined as the movement of something across the membrane in which no energy is required, and the item that's moving across the membrane is moving in response to what we call its concentration gradient. So the concentration gradient of any particle, any molecule, or any atom is that all molecules will always passively, without energy, naturally on their own, tend to move from their area of higher concentration to their area of lower concentration, meaning where their concentration is higher, we call that the high concentration area, and then somewhere else there's a lower concentration of that same molecule or atom, and that would be their low concentration area. And so diffusion is the specific name for the movement of molecules. Diffusion, though, doesn't have to be across a membrane. It can just be any movement. So we'll be talking about passive transport, where we're going to have something moving across a membrane, but it's going to be moving passively from its area of high concentration to its area of low concentration across the membrane. Now, in the picture they give you here, there's no membrane. So this is just simply diffusion. If you drop red dye into a beaker of water, the dye, when it first enters the water, that's the area of high concentration, right where the dye is. But very quickly it will move out in all directions because the pure water is the area of low concentration of dye. And eventually it will be equally distributed in the water. This example, though, is very theoretical because the weight of the molecule will have an effect on this if you actually did the experiment. But it's more of a mental experiment in that the concentration is highest at first, and then it will spread out through the beaker and become equally distributed. So we say that diffusion, the movement of molecules from their area of higher to lower concentration, will continue until there is no gradient. There is a uniform distribution of molecules throughout the whole liquid, let's say. So those are just some rules that are governing the passive movement of molecules with no output, no expenditure of energy to make it happen. So membranes, you'll remember, have that hydrophobic core section. That's that part where the hydrophobic tails of the phospholipid bilayer create that barrier. And so that is a major barrier. It repels polar molecules. So polar molecules cannot cross that barrier without the help of a pore protein. But nonpolar molecules can actually move across a membrane without any help. So they don't need the help of a pore protein, and so we say without facilitation. Facilitation means help. And so nonpolar molecules, and the ones that are important for us in this course would be like carbon dioxide is nonpolar and oxygen is nonpolar. Oxygen, that makes sense why it would be nonpolar because it's two oxygens, so they're equally sharing the electrons. The reason carbon dioxide is nonpolar has to do with the shape of the molecule. It's a linear molecule, and when you take chemistry, or if you've already taken chemistry, the shape of the molecule has an impact on the polarity, which we haven't considered in this course. But given that carbon dioxide and oxygen are both nonpolar, and also small lipids, these kinds of things can cross through the membrane with no assistance. They just cross over. And that's good news in general because oxygen can come in or out, carbon dioxide can come in or out. There's no dependence on any kind of pore proteins. Now, small polar molecules, and what I want you to think about when we think about small polar molecules, The smallest one is going to be water. But also think about your monomers, like your glucose is a decent size monomer, an amino acid is a monomer. So some of those monomers, and then things that are smaller would be ions, because those are just charged atoms. Those can cross the membrane, but only with facilitation. So facilitation means the help of a channel protein, one of those pore proteins that I showed you that creates that barrel or that pore shape through the membrane and can allow the passage of some of these polar molecules. So small polar molecules and ions need facilitation. Nonpolar molecules don't need any facilitation. So that's called simple diffusion. And then when facilitation is needed, that's called facilitated diffusion. In both cases, they're diffusion if the molecules are moving from their area of greater concentration to their area of lesser concentration. Now, the facilitation of the movement of these small molecules using the pore proteins has a size limit. So large molecules like polymers, polysaccharides, large proteins, these can only cross the membrane through a different process called bulk transport. And we'll look at bulk transport in the last lecture segment of this chapter. Okay, so let's talk about facilitated transport. So this is the molecules such as ions or the monomers like glucose or single amino acids. They cannot cross the membrane directly, but they can move through these, what we call channel proteins or carrier proteins. These are transmembrane proteins, and they facilitate the movement of other molecules from the outside environment to the inside, or from the inside back to the outside. So small molecules will move through these transmembrane channels and transmembrane carriers. So because these channel proteins and carrier proteins are very selective, and what we mean by selective is they don't just let anything pass through. There has to be a particular, there's a selection for particular molecules that are allowed through. We say that the cell membrane is selectively permeable. So I'm going to show you a picture. And in this picture, what they're trying to indicate to you in these diagrams is what the gradient is at the beginning of this process. So on the left-hand side, let's say, they're trying to indicate that there's more of this particle. This could be glucose, this could be an amino acid, it could be an ion. There's more of this particle in the extracellular space and less of this in the intracellular space in the cytoplasm. And so based on that, diffusion would move the particle this direction across the membrane from its area of higher concentration to its area of lower concentration. And this process on the left-hand side will stop when there's an equal amount of particles on both sides of the membrane. So they're only showing you the beginning setup, and then sometimes they'll show you the direction of movement, or you might be asked if this is a diffusion, which way would these particles move through this channel protein. On the right side, there's a different diagram. They're showing you a concentration higher on the inside of the cell, a lesser concentration on the outside of the cell. So diffusion will move these particles in this direction through the channel. So depending on what the question is asking, if they give you the arrow, they're showing you which direction of movement is happening, or they might ask you to draw the arrow or pick the right direction of arrow for a diffusion event. So diffusion always moves something from an area of higher concentration, and that can be either inside the cell or outside the cell, depending on the specific example. And then you can think of this process continuing until there's an equal amount of molecule on both sides of the membrane. It won't stop until it reaches that point. So this picture here is really depicting a channel. A channel is kind of a straight tube. You can almost think of like a tunnel. If you've ever gone through a mountain on a road, you go through a tunnel through a mountain. It's just a straight tunnel. The thing about a channel, though, is it's shaped exactly the shape of whatever molecule is supposed to go through it. So in that way, it's selective. So if this is a channel for glucose, then you can think of the hole that passes through this protein as being shaped exactly like glucose. So only glucose can go through that. Other molecules would not be able to fit through that hole. So channels are specific. I know when they draw them in the book, they look like anything could go through, but this is the way the artist decided to draw this, but in reality, they're very specific. So you have different channels that are dedicated to allowing usually just one or sometimes two molecules to pass through. All right. So they're not just wide open. Everything isn't just rushing through. So they're very particular. So this is an ion channel, and only a particular ion can go through that channel, for example. And then in this example, they're showing the diffusion. So that's what the arrow is showing. So this would be a facilitated diffusion through a channel protein. In this picture, this is a facilitated diffusion through a carrier protein. Again, this is a transmembrane protein. And in this diagram, what they're showing you from left to right is like a time lapse. So this is the starting time. When you first come into the event, this is what it looks like. Then this is when in the middle of the event, and this is the end picture. So it's like this time lapse, 1, then 2, then 3. So it's the same single carrier protein shown at 3 different time periods in succession. You could make an animation. I'm sure the book has one. So what you have here is this molecule, whatever this is, they show it as a purple square, will go into, it can only fit into the carrier protein that has the correct shape. But you'll notice here that the channel doesn't, the hole doesn't go all the way through like in a channel protein. And so this guy goes in, and then what happens is there's some kind of shape change. There's a shift, and it opens up on the other side. So I always think of this a little bit like a revolving door. If you've ever been in the airport, they have these revolving doors. And so when you go in, the door is open on the side that you're on, but then as it moves, then you have to go out on the other side. So it's not just a straight opening. You have to go in, and then it moves, and then you can go out on the other side. So that's the concept of a carrier protein. There's some kind of shape change, and then the item is released on the other side, and then it can do it again. It can repeat that whole process, bringing in molecules, as you can tell, from the side that has the higher concentration to the side that has the lesser concentration. So this is a diffusion, but this is a carrier protein simply because there's an indication in the artwork that there is some kind of shape change that happens as part of the process. Otherwise, a channel and a carrier aren't really that different. They both can move molecules across membranes. And if it's moving from an area of higher concentration to an area of lower concentration, then it's a facilitated diffusion. So in the next lecture segment, we're going to look at the movement of water across membranes. There are channel proteins that allow water to move across membranes, just like any other molecule, and they're very selective. And so when water moves across membranes, there are particular effects that can happen to cells when water is moved across membranes. So we'll pick that up on the next lecture segment.